Hello and welcome to Chapter 10 of Biology 2E. It's a kind of freeware out of Rice University, open stacks, I think it's called, with the primary editors of Clark, Choi, and Douglas. Chapter 10 begins with a section on cell division. Okay, lots of PowerPoints. It's a small section, lots of PowerPoints. So the cell cycle, I think that's actually the next section. But the cell cycle is the orderly sequence of events from one cell parent to two genetically identical daughter cells. So we're going to talk about mitosis, I think, Gesundheit in the next PowerPoint. But this one is on cell division. So what is a genome? Actually, by coincidence, I'm reading a book by Francis Collins, who is a Christian, who wrote this is a classic text now, even though it's only about 15 years old, called The Language of God. Francis Collins was in charge of the Human Genome Project, which basically mapped the entire human DNA cycle. So the genome is a cell's DNA. And of course, DNA is double-stranded, right? In humans, it's a double helix, right? You remember all that stuff from high school, right? And so the genome is it's like the map to an organism. If we have just in every cell of our body, in every cell of my body, there's a map to my whole body. In every cell of my body, there is a map that says he's going to have blue eyes and he's going to have brown hair until finally age takes over. My beard is white. It's strange. I don't dye my hair, I promise. But anyway, it's the map. And so the human genome is kind of like the way the whole thing lays out. So there's a spot on the human genome that says this is where hair color is decided, and so forth, and why I'm so handsome, all of that handsomeness is in the particulars of my genome. Now, the genome is the framework. It's not the specifics of blue eyes or brown eyes. It's the overall map that says this is the location where eye color is, and so forth. So the genome is basically the whole cell's DNA that tells you. So like humans all have 46 chromosomes, right? That's the human genome. And those chromosomes have genes on them, and that provides kind of the map to what a human being is as opposed to a chimpanzee. Of course, the human genome and the chimpanzee genome are very similar. I think there's only like 2%, and I'm sorry, I don't have a photographic memory. So it's like 98% of the chimpanzee's genome is the same as the human genome, and so forth. Okay, well, so the part of the cell where the genome is stored is, for somebody like me, it's in the nucleus. And in eukaryotic cells, it's in the nucleus. Now, the definition of a prokaryote is it doesn't have a well-defined nucleus. So we call it a nucleoid. It's nucleus-like. But anyway, prokaryotes, like bacteria and archaea, they don't have well-defined nuclei. And so that's what a prokaryote is, right? Prokaryote, pro means before. It's before things started having nucleuses. Why they let all their DNA just hang out there, those prokaryotes. But then the eukaryotes, the good karyotes, they had they put on underwear, the nucleus underwear that hid the DNA inside of it. Okay, so prokaryotes do not have nucleus. Okay, so the DNA in a prokaryote is a circle. The circle of prokaryotes. Anyway, circular molecule of DNA, and it's just a single molecule. We've got lots of molecules in our DNA. I guess you would say we have 46. Is a chromosome a molecule? Not 100% sure, but there's just a single circular molecule of DNA in a prokaryote. Okay, now a plasmid. This is mentioned both in the book and in a certain professor's notes, a certain professor's PowerPoints. A plasmid is like a little stray loop. It's like the black market for DNA. I've got a little plasmid. I got a little DNA I'd like to share with you. And actually, this is the way that that bacteria and so forth can get around the kind of human anti-immune system is by dealing in plasmids. Here, I can get you out of this um, anti what did I just call it? Anyway, I can get you out of the protections of the body with this little plasmid. So these are little stray loops of DNA that prokaryotes sometimes use, and these actually help them develop to where they can overcome things like anti-immune systems. That's the word I was trying to remember. Okay, prokaryotes. Bam, eukaryotes. These are the one with nuclei. These have a nucleus. And these are several molecules of DNA. Now, whereas in the prokaryote, DNAs tend to be circular, prokaryotes, in the eukaryotes, they tend to be linear, lines of DNA, and several molecules of DNA. And every organism has a number. Ours is 46. I'm not sure why they said 23andMe. Why did they say 23andMe? Because that's only half. And of course, a gamete, I don't think did I put gamete in? I don't know if I put that. A gamete is like half of our DNA. That's like in a sperm or an egg, because it's getting ready to meet up with somebody, So it's only got half. A gamete does. But in a normal cell of a human body, we've got 46 chromosomes. Okay, 46, that's our number. 46, that's the number for humans, number of chromosomes, number that we have. Okay, so there you have it. What's the difference between prokaryotes and eukaryotes? Seems like that's a study question somewhere. The difference between a prokaryote and a eukaryote is prokaryotes have circular DNA and just one molecule, whereas eukaryotes have linear DNA and lots of molecules. Okay, functions of cell division. Cell divisions for single-celled organisms, the function of cell division is reproduction, because now we've got two of these organisms. For multi-celled organisms like me, I'm a multi-celled organism. Growth, because if I'm going to get from being a little bitty baby, to a big Ken, there's going to have to be multiplication of cells. So growth is one function. Repair. Oh, this cell's damaged. We need a new one in here, so we reproduce the cells. The cell division then, provides us with healthy cells that aren't damaged. And of course, maintenance. Okay, so because, cells get old. I mean, sometimes I'm not 100% sure what the difference is between maintenance and repair, but you get the difference. Maintenance is something that keeps the same one going. Repair is something that fixes something that's broken. Maybe, okay. Well, okay, maybe I could, okay, I'm going to guess. Okay. So repair would be like I have a damaged muscle. So some people watching this video know that I hurt my right knee running because I'm getting old. It's very frustrating. I don't know whether I tore an MCL or what, but I'm trusting that for the last 2 months, there has been kind of cell division in that ligament to try to repair a ripped and broken MCL or whatever it is I messed up in my knee. So that's repair. But maintenance is, I can't have the same cells forever in a good muscle. And so it's going to have to keep dividing so that I keep a good muscle going. Okay, I tried. That's my guess as to what the difference is between maintenance and repair. Okay, human reproduction. So human eggs and sperm are a gamete, a half chromosome, 23 and me. So human eggs and sperm are half chromosome, a gamete. I put it in here. It's in the PowerPoints. So a gamete is a half DNA. We call that a haploid because it's half, one N instead of two. So that's a gamete. And they come together, two gametes, sperm and an egg, half and half, to form a zygote. A zygote is a diploid. Di meaning two. So that's 2M. Okay, stuff you probably remember from high school. Homologous chromosomes are matches made in heaven, where they come together. Okay, and they are one. We are one in the bond of chromosomes. Okay, human reproduction. Genes, okay, so if we were to take DNA and we were to unravel it and hold it out, the various places on the chromosome, various places are called genes. Genes are little segments of the chromosome that specify, they tell you which proteins, basically specify hair color, eye color, those sorts of things. That's a locus, a place. That's Latin for place. Locus is a place on the DNA chromosome that says hair color here. Okay, there you have it. So human reproduction. Okay, so DNA is long. So if you were to take a single DNA and stretch it out, it would be about 2 meters long. And so that's a long. Even though it's only 2 nanometers, that's 10 to the minus ninth wide, it would be 2 meters long. Well, all of that has to fit in a cell that's only 10 micrometers. That's 10 to the minus 6. So it would take 100,000 cells to stretch out into a meter. So these are really small. So how are we going to fit 2 meters in 10 micrometers? How are you going to do it? Well, we're going to do a little bit of wrapping. And so the DNA has to be wrapped tightly into the nucleus for it to fit in the cell. So what happens is that you wrap it around. So this is called a histone. A histone. Well, each one of these is a histone. There should be 8. I don't know how many there are here. I see 5 colors maybe, but there's supposed to be 8 proteins. This is called a histone octamer. Oct means octon. No, that's wrong. Oct means 8. So octamer is 8 proteins. So I don't know why it does this, but basically these are histone. They're called histone proteins. And so in the smallest level, we take a little DNA, and we wrap it around. We wrap a little bit of DNA around this octamer, this histone octamer. We wrap it around, and so we're trying to wrap it in so that we can cram it in the nucleus. Okay. So stage one of packing. Stage one of packing in the DNA is we take a histone octamer, 8 proteins, and we wrap a little bit of DNA around it. Okay, wrapping it. Okay, that's our first compaction. Second this is called a nucleosome, by the way. A nucleosome. A nucleosome is one little bead of wrapped nucleus, of wrapped DNA, one little bead of wrapped DNA around a histone octamer. Okay? Next, we take, so you can see that these are so this is a blown up version of this little bitty place right here. Okay? So you take all of these nucleosomes. This is compaction. We're compacting the DNA so that it can fit in the nucleus. First level of compaction is the nucleosome. Okay, we take histone proteins, 8 of them, an histone octamer, wrap a little DNA around it in a bead. That's the first level of compaction. Second level of compaction is that we kind of wind it into a coil of chromatin. This is called chromatin fiber. So each one of these little things is a nucleosome, and we coil it, and we connect it by what's called linker DNA. I don't know. I mean, so these junctures, I guess, is linker DNA. It's linking. So we got a little nucleosome, and we're gluing it together with linker DNA, and we're winding it into a coil, which is called a chromatin fiber. Now, it's about 50 times shorter than it would have been before by this compaction. Second level of compaction. The third level of compaction, we're going to pack the chromatin. So again, this little spot here is this big spot. All of this is in this little box right here. And so we further compact it by using other proteins. I guess these are supposed to be protein fibers that we're kind of packing the chromatin around the protein fibers. Okay, so we have packed in the DNA. This is a chromosome that's already been replicated. But anyway, more on that in a second. But you can see that this is just a little bitty spot on a chromosome. Okay? So what do we do to pack it? First, we took a little DNA and we wound it into a nucleosome around a octamer of histone octamer. We wrapped it around, a little bitty. This is DNA. There's a little bit of DNA right there. And we wrapped it around and we linked it together into a chromatin fiber. And then we wrapped that around more protein fibers. Okay, so that's how we're going to pack the DNA into a nucleus. Okay, so now DNA replicates. When does it replicate? It replicates in the interphase, so in between the actual cell. So you've got the cell division, which is the dividing of the overall kind of cell. But in between, this DNA replication takes place in the interphase, in between the phases of mitosis, in the S phase. Now, it's not technically part of mitosis. Mitosis is when it actually starts pulling apart, but it's getting ready for mitosis in the S phase by replicating the DNA. And so when after replication, you're going to have two identical chromatids joined together at a centromere. So this is after replication. This is a chromosome. We've got two chromatids. Okay, this is replicated. So these are, I think, identical. And we've tied them together in a little bow, getting ready for cell division. The center where they connect, where they're tied together, where they're glued together, that's called the centromere. Okay? And those are held together by a protein called a cohesin protein, which should be fairly easy to remember because it's cohesive. Okay? And that's this first section. There's a lot there. So, what do we need to know? What do we need to know from this video? We need to know the difference between prokaryotes and eukaryotes with regard to this. Prokaryotes have no nucleus and their DNA is circular and it's one molecule. The eukaryotes are more linear and you have multiple molecules, for example, 46 chromosomes for humans. This is the reasons why they divide. They divide to, if you're a single-celled organism, they divide to reproduce. If you're a multi-celled organism, they divide so you can grow, so that you can fix tissues that are damaged or maintain tissues that are okay. Human reproduction, you have these things with half the DNA. And then the DNA is packed by taking histone proteins and wrapping around a little bit of DNA around it. This is a histone octamer, and all wrapped together is a what? It is a nucleosome, okay? Then we're going to take the nucleosomes and we're going to kind of coil them into chromatin with an N, chromatin fibers. And then we're going to connect that to more proteins. And then when we replicate, it's in the interphase, in the S phase, in between. And this is a chromatid. So the chromatin fiber is how it's compacted. A chromatid is half of a body, half of a person, half of a chromosome, after it's replicated and gotten ready for cell division. And the centromere is what glues them together, and the protein that glues them together is cohesin. Okay, I did my best. This is the first video in Chapter 10 on cell division.